Let me introduce ourselves here. I'm Fred Reiss. I'm the chief architect at the IBM Spark Technology Center. I'm also a committer on the Apache SystemML project. I'm here today with Mike Dusenberry, my colleague from the Spark Technology Center, who works as a machine learning developer there, and who is also a committer on the Apache SystemML project. Just for some background, the Spark Technology Center is an open source lab run by IBM in San Francisco. We're located down on 505 Howard Street, if anyone wants to come by and visit sometime. We also have a website, spark.tc, to find out more about what IBM is doing with Spark and what the Spark Technology Center is doing with the Spark ecosystem. You can go there and find lots of information. Our mission as a center is threefold. We contribute intellectual capital and technical IP to the open source community, the Apache Spark open source community. We help to make Spark and its related ecosystem components more enterprise ready and to get them into IBM products and services. We also build data science skills at the Spark Technology Center, and we have a number of data scientists on staff. Now, the project that I'm going to be talking to you today about, and that Mike is going to be going into detail about today, started out as a suggestion from Mike and from Madison Myers, one of our interns from UC Berkeley. They came to me one day and they said, we have a great idea. Let's go and use Apache SystemML and deep learning to work towards addressing a socially relevant data science problem over big data in the healthcare domain. And this really pushed a lot of buttons with me because it's so nicely aligned with our mission. We're contributing to the capabilities of Apache SystemML, which is a system, an open source system for doing scalable machine learning, bringing that into the deep learning domain. We are helping to make the technology of deep learning more enterprise ready, which is of course strategic to our employer. Healthcare is a big business for IBM, both in traditional IT and also in our Watson portfolio. And of course, we're building data science skills and extending our existing data science skills into the deep learning domain with this project. So I was really excited to start this project off, and we've been working on this for a couple months now. Now, to work on healthcare, you need, of course, healthcare data. And being an open source lab, we like to work with open data. Working with open data helps us at the Spark Technology Center to share our work, share our methods, share our code with people outside of the center and have them be able to freely download the entire solution we put together and try it out, extend it for themselves. And the particular set of open data that we chose to work with was the data from the Breast Cancer Tumor Proliferation Assessment Challenge, TUPAC for short, 2016. You can get that from the website listed down below. This data set consists of about 500 images that look like this. And these are whole slide images. So the process that went into producing these images took 5 steps. First, someone took a biopsy of a cancerous tumor and then sliced that biopsy into wafer-thin slices and put each slice onto a slide for a microscope, then stained the slide, then stuck that slide onto a special high-tech microscope with an integrated camera with a stepper motor to drive the platen, and took photo after photo after photo after photo after photo at very high magnification of that sample. So once these images are digitized, typically you'll send them over to a pathologist who will examine them by hand and come up with a tumor proliferation or a cancer level score. In this case, we were dealing with the tumor proliferation score, which tells how fast the tumor is growing. So here's an example image, the same image you saw before, a little smaller here, so we can show some key stats. The key thing to keep in mind here is this is a 2.6 gigapixel image, because it's a composite of hundreds of different images. You can see a little bit of the borders between the images at the very top where those white lines are, that were taken step by step by step by step by a mechanized microscope setup. And just to give you an idea of how really big this image is, about the kind of scale that we're dealing with. And keep in mind there are 500 of these. Let's zoom in a little bit on this. There we are. Okay. So this is the scale that a pathologist doing a mitotic count, looking at the individual cells to see what percentage of them are dividing or growing in this slice of time would be looking at. If you look at the upper right, you can see the original full-scale image, and there is a little tiny red rectangle embedded up there. That's the region of the image we're looking at right now. So a pathologist will look at this image, will examine the nuclei of the individual cells, see what percentage of the cells are dividing. That gives you the mitotic count if you take that over a fixed area of the slide. Now, that mitotic count produces, after you've thresholded it, something called the tumor proliferation score. This is a score on a scale from 1 to 3, where 1 is best for the patient and 3 is worst for the patient, of how fast this tumor is growing. This is one of 3 components that one would typically use in a clinical setting to quantify the severity of the tumor. The other 2 components would be tubule formation, which is basically at a broad level, at low magnification, what percentage of the tissue looks normal versus cancerous, as well as nuclear size and pleomorphism, which is basically at a more microscopic level, what percentage of cells look distorted and how distorted do they look in terms of their cellular structure. But this mitotic count is a very important component, and so we're focusing on that because it's telling how fast is the cancer growing, how invasive is it. So our goal that we decided on, based on what we had in this data set, the data and the labels that we settled on, was to predict the tumor proliferation score from this whole slide image by using some machine learning to analyze the image. Now, there are only 3 of these scores possible because it's a scale from 1 to 3. So we can treat this problem as a classification problem with 3 classes. Now, as it happens, there's some very nice recent work out of Stanford that uses deep learning and convolutional neural nets to predict the overall tumor grade, not the proliferation score, but the high-level grade of the tumor on a smaller data set. So we figured we could borrow many of the same techniques here and power this up to predict on a much larger data set that we had available, and instead of predicting the tumor grade, predict the proliferation score, which was the data for which we had human-curated labels. Now, you'll note here, as I said, that this previous work was using deep learning, so I'm going to pause for a moment and just explain what is deep learning. A good way to think about deep learning is if traditional machine learning is a hammer, deep learning is a nail gun. It's machine learning with power tools. And just like with any other power tool, deep learning gives you the potential as a data scientist to be much more productive, to work much, much faster and get much further into a given data science problem. But like any power tool, it also opens up new and exciting ways to, so to speak, shoot yourself in the foot. And you want to avoid those pitfalls. So if you use traditional machine learning to do the image classification problem, and remember, we've formulated our problem as an image classification problem, the process would go roughly like what's on the screen here. You start out with a lot of raw data, images, and you start by massaging that data, cleaning it up into a format that is amenable to further analysis. Typically, after data cleaning, you have not image files, but arrays of integers. Then from this cleaned and reformatted data, you extract low-level features, things such as edges. And then from those low-level features, you can build up higher-level features like shapes. At this point, one typically has way too many features relative to the number of labeled points, so you include some dimensionality reduction at that point in the pipeline, and then you can start training a model like a support vector machine or perceptron to do the actual classification over that dimensionality reduced data. So that's traditional machine learning. At the end of the process, after some cross-validation, you get a model and some goodness of fit metrics. And typically, the first time that you do this as a practitioner, as a data scientist, you'll find the goodness of fit is nowhere near good enough for the application. So you have to go back and tweak the individual stages, maybe add new code to the individual stages. And usually the work is concentrated on the feature-related, feature extraction part of this pipeline. And so we call this feature engineering. It's a very time-consuming manual process. We got all the pictures in the back there? Great, okay. So now with deep learning, the picture changes. We take this manual feature engineering out of the picture and do that feature engineering task with power tools. And in this particular case, the power tool that we use is gradient descent, otherwise known as hill climbing. We use gradient descent in this case not to do the data cleaning, but to cover the other tasks in the machine learning pipeline. Feature extraction, dimensionality management, and of course the modeling itself. Now, in order to be able to express these steps in such a way that you can apply gradient descent, you need to be able to compute a gradient of this goodness of fit metric with respect to all the parameters that define those four processing steps. And in practice, what that means is these four processing steps become a chain of neural networks, which taken together become a single deep neural network. So in principle, once you've reformulated your processing steps as a neural network, the automated machinery just comes in there and you no longer need data scientists. In practice, though, that's not exactly the case. Because again, this gradient descent, it's a power tool. It will apply a great deal of brute force in exactly the direction you point it at. And it's very important to point it in the right direction, and there is always someone behind the scenes constructing the neural network, constructing the data cleaning pipeline, so that when gradient descent is applied here, you end up with a good model. If you don't properly construct everything, you end up with the major risk of working with deep learning, which is overfitting. Now remember, we've got this big neural network in place of the last four stages of processing. And in effect, what that has done is instead of having a small, constrained modeling phase, we have a gigantic model which is going to have millions or billions of parameters, and you're going to have orders of magnitude fewer labeled examples, and that is a guaranteed recipe for overfitting. Overfitting means that the machine learning components are going to discover correlations that just occurred because of pure random chance, because there are so many variables. And the way that you avoid overfitting is with a process called regularization, which basically means smoothing the search space for the model, restricting that search space so that areas of that search space that contain a lot of these false correlations are cut off. Traditionally, you would do regularization with a regularization parameter that governs how high the sum of the model coefficients would be, or the sum of squares. In the case of deep learning, you have to be much more aggressive and much more creative about doing regularization, to the point of using techniques that you wouldn't normally think of as regularization to regularize that search space. So for example, you might change the topology of the neural network or freeze parts of that topology so as to prevent them from finding bad correlations. You might, if you find that there are certain classes of correlations in your input data that are just leading to problems and prediction, you might add noise to the input data, randomly perturb images so that you cancel out those problematic correlations. So lots of tricks that you need to apply here, all about doing really creative regularization to avoid overfitting, because that's the part where this power tool can sort of shoot you in the foot. But again, it's a very powerful technique that has allowed a lot of practitioners to get really, really positive results. So to talk about our results and the tools that we use, I'm going to now hand things over to Mike.
All right, thank you, Fred.
Sure.
All right, can you guys hear me? Excellent. Okay, so quick recap, right? Just to bring us back to where we are. At this point, we've formulated a problem in which we want to take whole slide images, extremely high resolution whole slide images, and map that out to a tumor proliferation score. And we phrase this problem as a classification problem, right? So first, let me go into the tools we use, and then let's jump into the whole process, right? So pretty quickly, we used Apache Spark, we used Keras, we used TensorFlow briefly, and we used Apache SystemML. So Spark gives us kind of large-scale data processing capabilities and lets us scale out our preprocessing pipeline and also run our engine for SystemML. Keras is a fantastic API for defining neural networks, and so we used it for that. We used TensorFlow as a backend initially for Keras, while we were also working on advanced features for the engine inside SystemML for deep learning. And then we used SystemML as a backend at the end for Keras for scaling up our deep learning models. Right? So let me quickly talk about what SystemML is now. So Apache SystemML is a project for large-scale machine learning in a distributed environment, right? So essentially what that means is that the project exposes high-level languages in either R-like or Python-like flavors. And essentially that means we pull out the linear algebra portions of these languages and expose them to the user. And so the user can write their algorithms in the form of math and then supply hardware and will automatically scale up the algorithms. And we work pretty closely with Spark. We also have a Hadoop backend. But for Spark, if you can invoke Spark, whether it's via Python, Scala, or Java, you can also pull SystemML in as a library and immediately scale up your algorithms. And it is an Apache project, and we're in the Apache incubator phase currently. So let's talk about it. So we talked about this pipeline, right? And so traditional machine learning has several steps, perhaps you could think of it. And deep learning or neural nets kind of consumes part of that pipeline, right? But as we noticed, right, the kind of data prep or data cleaning section is not part of that, right? So let's go into that first. Figure out how did we take our problem. Because if you think about neural nets, it's pretty common to have a large amount of data. You're thinking millions of examples, but we only have 500 examples, right? So this is where you try to pull in some of the domain knowledge that you understand to see, can you generate more data? In our case, right, we have these extremely large whole slide images, and they're biopsied from a tumor. And in practice, a physician will go in with a microscope and examine multiple areas at high resolution, and then decide an overall score. So they're going to use multiple areas of the image. And so they kind of assume that most of the image is the tumor. So we'll make the same assumption. So our hypothesis here, and we pulled this is inspired by the paper that we referenced, is what if we chop up the whole slide image into tiles? We'll chop it up into tons and tons and tons of tiles. You saw how large it was, right? And we'll extract out these tiles. And these tiles will be at 1024 by 1024 pixels, which is at 20x magnification is a medically relevant size that a pathologist would use for certain types of tasks. And if you notice, too, that the grid is not to scale. If that was actually to scale, it would be much smaller. So this is interesting because this allows us to take our relatively small 500 slides and expand it out to a much larger number, right? So at this point, we're ready to go, right? We're done. We're ready to plug it into a neural net and be done, right? Not quite. So if you look at, say, the top center of the image or the bottom right of the whole slide image, you'll notice that it's blank. There's just not any tissue there. As you recall, right, these are randomly shaped pieces of tissue laid on a slide, and so some areas of the image will just be blank, and we don't want that, right? So we did some filtering, right, as we can't just pass in blank images to a neural net. And so, this is inspired, we won't go too much in depth, but inspired by the paper, but essentially, we want to pull out areas that just have tissue. Simple way would be to say, okay, if the background's white, we'll just get rid of that. Problem is the background's not always white. It's some shade of gray or even darker than that, depending on shadowing or other characteristics. So we kind of empirically went through this, and we found a quick algorithm that basically looks for edges in the tissue. We assume no matter what the color of the background is, it won't have any edges, whereas tissue will have edges that define the areas around the cells or other stroma inside the tissue. And so we were able to take the tile in a high level, turn that into a grayscale, detect edges, smooth those edges out to determine what relative area is held by tissue, and then take a percentage. So the images, top left shows the original tile in this case. Top right shows the tile in grayscale. The bottom left shows the edges. We've pulled out edges, and this entire tile has tissue on it, so there's edges everywhere. And then the bottom right shows us smoothing it out. Any black region would represent tissue. For this slide, that's all tissue, so we can safely say this is a tissue slide, and we'll keep it. Right. So now we've got a set of tiles. We filter them, and now we're done. Right. Now we can plug these into a neural net and be finished with the whole problem. Once again, still not quite. Right. Even though this is a medically relevant size, 1024 by 1024 is still quite large for today's hardware in terms of either hardware memory or time constraints. There are models that can scale up to this for prediction, but for efficiency during training, it's much better to have a smaller image. So we actually, this is similar to the paper as well. We take our tiles, and once we filter them, we chop them up into smaller samples. And the samples are sized at a resolution and a size of 256 by 256, which is pretty common size for the neural nets in practice today. So a lot of the benchmark data sets, like ImageNet, are roughly around the same size, and so it works out nicely. So we've got this pipeline done. It's in Python because there's a high performance library for reading this special type of image format that the whole slides come in. There's NumPy. There's other libraries that make this type of work possible. But we don't want to wait for it to run on a single machine. So we use Spark to scale up the data processing. And this is about all the code it took to take our existing single node Python implementation and scale it up with as many nodes as we had. So essentially, I won't go through the code, but just making use of maps and flat maps and other high-level API features that Spark gives you allows you to scale this up. And so really drive this home, our 500 slides turned into 4 million examples. So at this point, we have 4 million labeled examples where we take the original slide, chop it up, and if the original slide had a certain label, the corresponding sample will now have that same label. Right? Because this is what a physician does. They look at certain areas of the slide and then overall give it a score. So let's shift gears a little bit now. Right? Now we've got our data set. We're ready to go. So now neural nets. So state of the art for this type of problem, kind of image classification problem, is a convolutional neural network. Right? So this is a class of models, just like a neural net. It's a class of models. And we won't go into details of what that is, but essentially a convolutional neural network as compared to a neural network or other type of model. It's just a model that takes into account spatial structure, so it works quite well for images. While you can build these up by scratch, that's kind of mindset of deep learning is build from small building blocks. If you're planning to apply it to a certain problem, it can be helpful to start with a model that's been built by someone in the community and trained on a benchmark data set for which there exists the math, a model implementation, and even the weights that were trained in the experiments for which the research was done. If you start with that, you can manipulate that model and then massage the weights for your particular problem. And so talking earlier about regularization, this lets you take a model that's quite large and that's very powerful, but start it at a good initial starting point. And by only training certain portions of it, we can minimize overfitting, but still manage to get the benefits. So what does that mean? So we get a library, and we've got this, in our case, we used a ResNet 50 model, which is a residual neural network with 50 layers. This diagram is of the 34-layer variant, but they're quite similar. And if you grab this out and you grab it with the pre-trained weights, you'll find that the task for which it was trained on was ImageNet, which has 1,000 classes. We have 3 classes, and they're not cats and dogs and buildings, right? So we start with a pre-trained model, but we want to rip off the end. We want to rip the 1,000 class classifier off and replace it with our own new 3-class classifier, right? So we replace that. And then we should train, right? Not quite. Once we replace that, we would be starting with a random end final layer, right? We don't want to destroy the rest of the weights while we're trying to get that search for good parameters for that final layer. So the common practice for this is to freeze your existing model. So you've got these pre-trained weights you start with. You don't want to destroy them quite yet. So you freeze that model, and then you train just the new classifier you've placed on top of the model. Right? So we're kind of doing some surgery on a pre-made model. And this lets you get at least the final layer into a good search space. But after that, right, these models, right, we're using pre-trained models because we're making the assumption that a model that's trained on some image classification tasks will learn at least lower and mid-level features that are applicable to lots of images, lots of image tasks, such as edges and shapes and other such things. But high-level features may be more inclined to be related to the actual classification task that it was trained on. So the higher level features may be more related to cats and dogs and birds for ImageNet. So the practice for that is to, once you've kind of trained this final layer, you peel back some of the frozen layers. So you unfreeze some portion of the network. Once again, we don't want to do the whole thing because we don't want to overfit immediately. These are very large networks with lots of parameters. But we do want to try to massage it for our problem. So we unfreeze part of it, and we fine tune. So in other words, we train this last piece. We train it very slowly, because once again, we don't want to destroy the weights that were already there. We want to kind of just massage them for our problem. So what does this look like for Keras, right? So we use the Keras. Once again, it's an API for defining neural networks. The Keras API, they have a ResNet 50 model. There's nothing magic. You can look into the file. It's very clean and simple to see. This is the code you need to grab a 50-layer ResNet model with pre-trained weights and with your own 3-class classification layer at the very end. That's all you need, right? And like we said, we want to freeze the existing layers. So there's just a simple trainable Boolean. We freeze them. We compile the model. We train the model, training just, at this point, just the final layer. And then we unfreeze some small portion. In this case, we unfreeze the final 2 residual blocks, if you're familiar with the ResNet. We recompile it with a slower learning rate, because we want to just massage the parameters at this point, and we train. So all in all, there's not that much code to do this, which is nice. It keeps it simple and allows you to focus on the math. But it's also extensible, so you can get into it and make modifications if you need to. And so initially, when we ran this on Keras, right, we were working on the engine, working on advanced features for the engine SystemML. And so we were kind of doing both things at once. And so initially, we ran Keras using the TensorFlow backend, which is kind of its default backend. There's also Theano. But then, once we had worked on some of these more advanced features in the engine SystemML, we switched over and placed SystemML as a backend for Keras and started scaling up our models. And so the nice thing about this is we had nodes available to us. These are the same nodes we were using for all the Spark preprocessing. And so by throwing more nodes at it, we were able to get a higher processing rate for our model. So you can kind of see some either the time or processing rate on the right that, with 3 nodes, perhaps it took 15 hours for this given experiment. We bumped up to 6 nodes. Now we've got 7 hours only. And up to 9 nodes, we're down to 5 hours for this experiment. So that means is you have more time to focus on your problem, and you can iterate quicker by just throwing hardware that you may already have at the problem. And so we found, too, is we wanted to compare, well, what's the difference between, say, a GPU-powered machine, which are a little bit harder to come by, and just kind of commodity servers that a lot of companies will have. For our experiments, we found that 1 GPU is about worth 4 machines. So the 1 GPU does great, but if you want to scale it past that, they are expensive and they're hard to come by versus kind of the servers that you most likely already have. So that was great. And so SystemML, there's not much to it from the ML part, which is the idea. We want you to focus on machine learning, and you bring the model you have, you bring the hardware, and we'll scale it. That's the goal. So let's tie it all together, right? So here's the overall pipeline. So once again, we start with the whole slide image. These are high resolution, maybe 2.6 billion pixels. We decide to tile it up, chop it up into tiles so we can generate more data. We filter out all the background, and then we chop those up into samples that are more amenable for time and hardware constraints. Each of these samples now has a label corresponding to the label of the whole slide. Pass these labeled samples into a ResNet, and we train it piecewise like we discussed. And just to point out, too, that we did split data from training and validation sets based on the original slide number. And so then you would have maybe 400 slides in a training set, 100 slides in validation, and then you'll do this whole pipeline with each set. All right. So let's look at some initial results and kind of talk about some interesting findings we had and pitfalls, like we were saying, like make sure you don't shoot yourself in the foot as you're doing this. So we want to look at first, we'll look at a way of evaluating how well our model did. So there's class 1, which is the best prognosis, and class 3 is the worst prognosis. So we said, well, let's look first at how well our model is able to distinguish class 1s from class 3s with maybe some ambiguity for class 2. So if a model sample is actually a class 1, what's the chance of predicting either class 1 or class 2? The idea is definitely not class 3. So for training, we were 98% accurate, right, which is fantastic. Validation, 76, maybe not so much. So let's see for class 3. If we at least do a 2 or 3 for class 3, definitely not a class 1. Let's look at the score. So for training, about 98.6% accurate. Doing great again, right? Validation, we're down to 60% for this, so that's not great. If we average that together, right, we're at 98.5% for training and only 68% for validation. So overfitting, right? But it's an interesting problem because, for deep learning, one of the initial things you would think of is, well, how about more data? Well, at this point, we have 4 million samples, almost 3 million in the training set, and say ImageNet only has about 1.6 million for their classification task. So maybe not data. What about overfitting with your model? Your model's just too expressive. Well, we started with a pre-trained model, so we have a good initial starting point, and we've limited by freezing most of the model, so we're not actually allowing the model to change most of its weights. We're forcing it to learn based on weights it's learned from different tasks. So maybe not that either. So this goes back to the idea that neural networks don't cover the data prep part. And so if you come across a problem like this, you have to really think about the data and the domain. So we got back into the data and we started looking, and we found that although we were getting rid of background tiles when we were filtering, we found it wasn't perfect. We were getting a lot of false positives. We were getting tiles that had tissue, but it was mainly junk tissue. Right? So it's either just noisy tissue or very lightly stained, particularly it's at the peripheral edges of the whole slide. So either the noisy stroma or adipose tissue or fat, which is mostly clear but has a well-defined border and could trick our algorithm into thinking it's tissue. So we said, okay, let's get more aggressive and filter better. And we won't go into how we did it. We just added additional checks. And once we did that, we found that our training accuracy almost didn't change much, a little bit down. The validation started to go up. So that's good, right? But less overfitting, but still overfitting. So, we were making some progress. So then we started really thinking about the data again, right? And trying to think about the domain and how this data is generated. So when a pathologist stains a slide, he or she can choose to put more stain or less stain. And more stain or less stain might result in a slide that's either more vibrant or less vibrant in colors. And so we started thinking, well, our samples, even though we have millions of them, they come from only 500 original samples. So if the original samples had distinctly different looks just based on having too much or too little stain, our network, which is very expressive, could simply learn which whole slide the sample came from, and then it would just have to split on 400 slides. So it could learn a much smaller problem, but not actually learn anything. We found that in the literature, this was a common problem, and there were solutions for it. So you find, hopefully you can see here, that these 3 slides kind of display different characteristics, even though they're all stained essentially with the same pink and blue stains. Whereas with some normalization, they become much more similar in quality. So we found a paper. This was also referenced by the dataset source. And we won't go into it, but essentially you take an image, apply some linear algebra, including eigen decomposition, and out comes an image that will be same appearance visually as other images. So it kind of normalizes the stain colors. And so not only visual, but also if you take a mathematical mean, you'll find that the means of these images, like pixel mean, is much closer now than it was before. So we did that, and we find our training accuracy goes down again, which we want, right? We want it to actually be trying to learn a harder problem rather than an easier problem. Our validation is going up still, right? So this is essentially regularization by actually cleaning the data and thinking about the domain when we work with the data. And so overall, you can see, by simply making more intelligent decisions on the data and not just trying to throw more power in terms of modeling, we're able to actually get increases in our validation score. So in summary, right, we made this kind of an analogy that deep learning is like machine learning with power tools, right? And so these are extremely powerful models with very high expressivity, but you still need to provide guidance to them. Even though they compose a large portion of the existing traditional machine learning pipeline, you still need to think about your classic machine learning techniques, and you think about the domain carefully, and with guidance, you can really work well with them. Domain adaptation is hard, but like I said, understanding it can help you to be able to work better with the actual machine learning. So in our case, we only started with 500 slides, which would, for most problems, not be enough. But understanding the domain, understanding where the data came from, allowed us to generate millions of samples that we could train with. And then later on, actually understanding what tissue, what do we actually want to see, and as well as understanding stain characteristics allowed us to actually increase our accuracy. SystemML. So SystemML allows us to scale up these kind of jobs, right? So we started with Keras, which let us think about the machine learning. And once we were ready, we were able to use SystemML to scale that up to the available hardware that we had, which was nice. And And finally, just want to kind of reach out and say, please come collaborate with us. Being part of the group that we're with at IBM, namely the Spark Technology Center, we're uniquely positioned to be able to work in the community and work in open source. So we have this code up on GitHub. We have the data is available, and we'd love to work with you. If you're interested in working on this breast cancer problem with SystemML, we have it. If you're interested in working on machine learning and deep learning in general, whether it's working with our algorithms, submitting your own or tinkering with ones we have, we'd love to work together. If you're interested in systems work, it's actually a lot of work to be able to scale up general machine learning to a distributed environment in terms of an engine. So we have lots of fun problems and interesting things to work on. We'd love to collaborate. Thank you. Yeah, good question. So the question was, have we rotated images in order to provide more samples to the model? So we did. So that would be kind of a form of data augmentation. For this talk, we just kind of talked about some of the initial things we ran into to have good examples. But yes, we definitely are working with data augmentation. So rotating, flipping, and also jittering the colors and brightness and saturation of the samples. So great question. Sure. Yeah.
Sometimes it might still be normal, right? Even for some with tumor slide, some tile might still be normal, right? So when you assign a slide label to that tile, do you think there's a problem?
So let me repeat the question.
Go for it.
So the question is, if when you're looking, our model is looking at individual pinpoint size segments of the original tile, and we're trying to label the entire tile. And some portions of that biopsy are going to be healthy tissue. Mm-hmm. Does that impact negatively the overall performance?
Yeah, so it's a great question, right? And that's where the domain knowledge comes into play. So from a medical practice thing, you're going to biopsy a tumor. So you're already assuming that the tissue is a tumor. However, you certainly could accidentally grab some tissue outside of the biopsy, and there is, for some types of cancers, we'll have pockets or regions that are more expressive of the tumor. So that's always a concern, for sure. You hope that there's enough tumor tissue overall from the slide to normalize over any other regions for it. But it's an excellent question that would come up.
Right. So statistically, at the level of the whole slide, you want to be able to take a number of possibly inaccurate predictions at the level of individual points and aggregate those together.
Yeah.
So another aspect we didn't talk about is when you consider the quality of the results coming from the deep learning model in this particular domain, you really want to look at how correlated the errors are. Because if the errors cancel each other out on average, you can leverage like the central limit theorem to put nice narrow bounds on the mean score that you predict for the entire slide.
Alternatively, you can do some kind of cluster analysis on the hidden layer output to see cluster pattern.
Yeah, we could do some clustering on the hidden layer output, or even change the topology of the neural net so it's got an explicitly two-phase classifier. Yeah, or cross products of classes.
Can you repeat the question, please?
Sorry, he was saying we could do some clustering on the tissue type on the so that we can essentially enhance our labels to help in training, which is a very good point. So.
I have a follow-up question. So, have you ever considered about treating Strata and Apache separate, not just
Yeah.
Did you hear the question?
Yeah. So have you ever looked at sampling from versus stroma versus actual cellular components? Is that right? Yeah. So in our case, we took just the whole slide image and chopped it up because this was kind of inspired by the paper. However, there is another way you could go. So oftentimes the pathologist will look at the slide also from a little bit higher resolution or less magnification, and will decide regions that they think are probably the worst, and then they'll zoom into those regions, there's several of them, and then they'll sample from that. So one way you could do it, and there is another auxiliary data set that goes along with our data set that has these regions of interest. And so one possible pipeline is you could add a region of interest identifier or classifier before our one. So you could identify the worst regions based on labels from a pathologist and then classify inside those regions. And that might help you to reduce any areas that maybe are normal. Great question. Yes?
Sorry?
Existing neural network, essentially the transfer of ResNet from Keras.
Yeah.
The choice of what you leverage come into play regarding overfitting, maybe a different kind of existing neural network versus the one you picked.
Yeah. So basically, we chose a ResNet 50. Would a different model employ better results or different results? Certainly, yeah. In our case, we just took probably what's one of the more state-of-the-art currently. Another possibility would have been like Google's Inception model. ResNets have pretty good results now, like very good results in the benchmarks. So we chose that. However, transfer learning itself hasn't been shown a lot yet on some of these newer ones. So there's certainly a possibility to explore with other model architectures and even get more into just custom-made ones. So great question.
So we've been told we're out of time. I think we'll stand out in the hallway out back if you have some additional follow-on questions. But thank you very much for coming to our talk.
Thank you.
Really appreciate it.